Okay, students, today we'll start with the topic of bacteria, but you must be seeing on your screens that it's written prokaryotes, right? But we have to study prokaryotes because bacteria are a subgroup of prokaryotes. Before we start with the topic of this prokaryotes and bacteria, I have to tell you certain things that before this, what we did was viruses, right? And viruses are, if you remember, what viruses are not non-living organisms, right? We call them particles. We never refer to them as organisms. And and the main body of virus, if you remember, if you just do a quick recap, the main body of the virus is called, the whole body, not just the main body, but the whole body of viruses are called virion. And the viruses they always need a living host to replicate themselves. And also remember that unlike bacteria, which cases one bacteria gives rise to one bacterial cell give rise to two other bacteria, which are called daughter cells, right? Uh, Unlike in the case of bacteria, where one mother cell gives rise to two daughter cells, in viruses, one virus can give rise to hundreds or thousands of virions. You understand my point? One viral particle, which we call virion, can give rise to hundreds or thousands of virions. So, four important points in case of viruses, that they are, the main body of virus is called virion. They are non-living, right? They are non-living. It doesn't, just the fact that they can replicate themselves doesn't mean that they are living organisms. Okay, they are non-living, and they are, the main body of virus is called, second, the main body of virus is called virion. Third, they always need a living host. They always need a living host to replicate themselves. And fourth, one virion can give rise to hundreds of thousands of virions, unlike in eukaryotic or prokaryotic cells. Now I just want to make one more point, because I found it really difficult to upload my slide or video on the YouTube, because they were getting too heavy in terms of memory. So what I will do from now on is I will make 10 to 15 minutes lectures, okay, and upload it on YouTube or any other, if possible, any other social media platform. And also I'll use a combination of Ladakhi and English language. I'm not going to, I'll make it clear, I'm not going to use Hindi, okay? English, because by the time I just hope that by the time you get out of this college or pass out from this college, you are at least able to, you are at least comfortable with hearing someone speaking in English, right? I mean, to be, I mean, mm, it would be much better if you people, when you passed out from this college, you are very proficient in spoken English. But I just hope that, because I have seen in the past that our students find it, they are not very comfortable with speaking in English. So what I'll do is, because you people find it very hard to understand, in, I mean, the this English language, you have problem with that. I mean, I'm not blaming you people, but because you have the problem, what I'll do is I'll speak in English and Ladakhi, a mixture of both. And now some of you might be asking, why not Hindi? Then I'll say, you see, Hindi is not our mother tongue, right? Our mother tongue is Ladakhi. And English, then you might say, English is not also our mother tongue. Then English is very important. See, English is the first business language. It's also the language of the science, and there's no way getting around this. There's no shortcuts around English. I mean, if you want to do good in life, you have to know English. Okay, So let's start with the topic, starting with the prokaryotes. Prokaryotes are organisms. I'm saying organisms. Okay. Now, for the unlike in the viruses case where we call it particle, right? Viral particle, or we say virion, and they are a virion is a particle. They are non-living, right? And prokaryotes are living organisms like human, like animal, like plants, any other living organisms. Okay, so they are prokaryotes are living organisms. And as I already told you, we are studying we I mean, I'm just giving you a brief introduction of what prokaryotes are, because bacteria is a subgroup of prokaryotes, right? So uh, prokaryotes, and pro means before. Okay, karyotes comes from the word karyon. Karyon means related to nucleus. Okay, so prokaryotes means before nucleus. Or so in the terms in the time scale of evolution, the prokaryotes appeared in biological time period, in evolutionary time scale. If we look at the evolution, prokaryotes appeared before the eukaryotes. Okay, fine. So prokaryotes are organisms that do not have nucleus. So if I speak the full definition of, coming to the full definition, so this is my logo. As you already know that I'm Stanzin Dadul. So, and so coming to the main topic, what we can do is, what are prokaryotes? So prokaryotes are any organisms that do not have nucleus, first. Second thing, they do not have any membrane bound organelles. Now I'm starting with the assumption that all of you know what organelles are, right? Okay, so if you don't know, you just go to the internet and check. That is an assignment for you, for those of you who don't know. ठीक है मेरा तेने ओते वेस्टिंग ना था prokaryotes po chin ok den naga to. Sorry, I'm going with the flow, and I'm only ngi ladak sirf English na spera ta ra tangi chi chun ngis ka mix choste ta ठीक ना मेरा ta ote bina prokaryotes na agar chin ok zen naga to, agar ngi English na zen naga to, prokaryotes are organisms that do not have nucleus and other membrane bound organelles. That is the basic definition. You can just write it down anywhere on your notebook and memorize it. Okay, no nucleus, no membrane bound organelles. Okay, so prokaryotes are divided into two other subgroups. One is bacteria. And bacteria, what are bacteria? Bacteria are prokaryotic single cellular organisms. Okay, they are, now remember, they are single cellular organisms without a nucleus and other membrane bound organelles. Okay, fine. They have non-membrane bound organelles like ribosome. And what are the non-membrane bound organelles? Ribosomes. Okay, and ribosomes are, if you remember, ribosomes are also found in eukaryotic cells, right? Humans have eukaryotic cells because we have nucleus. ठीक है मेरा तेने ओते वेस्टिंग ने second subgroup चिनोक जेना का तो आर्किया. ठीक ना मेरा आर्किया माने इंग्लिश ना वर्ड चिकाउं चन आर्काइक जते आर्काइक माने निंपा. ठीक ना मेरा आर्किया are also prokaryotic organisms and single cellular. Okay, ठीक ना मेरा, and they also have non-membrane bound organelles, and they non-membrane bound organelles. Was it? They do not have nucleus में क्या. ठीक ना मेरा, पर कौन bacteria ने कजू को सोसो इन जरना का तो, because they are, अगर ने genetically स्नाना का तो archaea गुन सब eukaryotes ना यहाँ पो सब एक शिक similar हो क्या. हाँ गोमा को bacteria बोंगा तंग ने eukaryotic cells ने माँ थागरिंग चौकशी को क्या. ठीक ना मेरा, तो यहाँ पो यू गुन archaea गुन earth origin जरा नोपा गाता है. If you, I mean, I don't know if you have studied the origin, anything about the origin of earth and evolution of life and origin of life. Okay, ta ive kanga ta religion is pera tangchas manok ki zuko god is kazuk siga create just no. Science has nothing to do, as much as I believe science has nothing to do with religion. ठीक ना मेरा, ओजर खोकोना यहाँ सोचे कि नो science ना religion ड्रेस गारिक माने contradict छानो ओजर. ठीक ना मेरा तेने Archaea un karu thobanok zenna kato. There are also some, mostly they are found in extreme conditions like hot springs. Kavena young organism gun dugnya na mera khala. ठीक ना मेरा ओजन mostly it's hot springs gun na mamang po thobanok archaea gun. Oje chiazana ngal nga nge daksa chilo archaea bacteria sangal la yuk lo amalo otebe jaste chi yuk na kato ngal nga ta agar stas te na kato earth temperature bu ma chante yuk ठीक ना मेरा otebe jaste then I mean archaeas are mostly found in very extreme conditions like very hot temperature, high pH, low pH yung chamana hagamago yamane marhante basic environment na habitat na kya yamane acidic habitat na kya ya temperature mane mamang po kya. For example, I just said the hot springs ठीक ना मेरा hagamago. Coming to the next topic, investing na chiut kya zenna kato ngata if we look at the relationship between bacteria, archaea, and eukaryotic cells, domain life a domain hala. Domain sorry, kingdom. Was it? Kingdom bo hala. Kingdom bo bacteria, archaea nang eukaryota. Was it? Take namera. Hagwa. Ote bena ikane chiduk zernakato. Ikane dugata. Ngala ane purple teudu gaminu purple bu chinok bacteria archaea bu du gaminu in red na hala tene eukaryota bu brown na hala ni circle chuste du gaminu ta ote bena chiduk zerna kato bacteria bu agar ni archaea astana kato bacteria ngala duk archaea sting na duk hala matlab chi song ive this phylogenetic tree shows that archaea is more similar to eukaryotes than bacteria. Hagomago bacteria ma eukaryot ne ngemoduga ya archaea ngemoduk archaea bu ngemoduk inna jugongis ka prokaryotes no he par nge cellular structure bo agar nge scientist ko ne detail in a study chostyot kyapa tebe nanga ta chi thons kya what we have seen is that archaea are more closely related to eukaryotic cells than bacteria. Hagomago thik na mera ta investing na ngata chi chonjen na kato we will come to the topic of bacteria ngata itu bor he archaea are where are the archaea found in extreme conditions extreme pH mane pH ya mane low either at very low pH or at very high pH ठीक है मेरा or at very high temperature where it's very difficult for other organisms to survive then a special adaptations होते क्या होते भी ठीक ना मेरा tang atang yung investing na we come to the bacteria ठीक ना मेरा hagwa ta bacteria ngata inna जुगे इउ ची नोक्जन ना कातो या इउ ta computer na ngata ma izugo bacteria mane ma demo chuste thoma no ngata ote wo electron microscope na staste agar nga pura structure bo ngata light microscope ka ngata lab gun na yo ote be na thano पर मा डेमो चुस्ते थमन तेबे फेंगाता ची चोगो सनक माने yang expert go सनक ते study चुचे से पिया हाला तेने खोंगने माने angle सो ने bacteria स्तास्ते तेने खोंगने ओतेओ decode चोनक ते हाला declutter चोनक या decode चोनक I mean and make it easy so that make it easier for us to understand हागो मगो पर ओन्ना हागो चेसे पिया scientist कोने कोरंग मने life पतांस्तानो हाला तेने इकाची दिस्ते a computer generated image of E. coli Escherichia coli zeremina hala ठीक ना मेरा इवंग CDC nginga lang zata mana Center for Disease Control USA hala ठीक है मेरा Alyssa Eckert na Jennifer uste reason iuchi no khongne scientist kune mingono kabe e structure bo computer nangem po ngala pura electron microscope nangem po study choste tene izuge image phingstano I raskut chokson nang chino gotegun flagella na cilia no oje sorry I mean cilia is generally found in the eukaryotic cells niskata flagella zertong ठीक ना मेरा या sex pili zertong या pila zertong a chun chun raskut kun du gameno pink po hala ote gun chino pila no agun ring mo gun flagella no oje ta I gun pila na flagella gun chino zerna ka to ngata oteo kule sele ta inna structure bo agar nge demek chuste zerna hala ata ibina chang teza chik sabik chik rough mane ta ibina pura label menu khala ta ngata ha go chese pe chi chon zerna ka to izu image chik du gameno E image pena chi duk zerna ka to stos he even this is a bacterial cell hagomako and small protrusions or projections are what pili they are called pili they are normally used for attachment to the surfaces like during sexual reproduction of bacteria hagomako nge chilo attachment it helps in attachment to the surfaces hagoma ko especially during sexual reproduction in bacteria hagoma ko te ba resgarek specifically khongna ya sex pili yang jana no resgarek hagoma ko sex pili subgroup chikeno and outer covering tene ote beka this is the longer threads are what flagella in ikangis tante to gamen flagella hala sorry flagella to gamen hala ta even I'll talk about this in details then I said basic definition of prokaryotes are that they do not have nucleus and other membrane bound organelles ठीक ना मेरा membrane सिर्फ जगह चिक चिका क्या होते हो खोए साइटो सेल मेम्ब्रेन ठीक ना मेरा चिका कजु गयो क्या लो सेल मेम्ब्रेन हाला इने ठीक ना मेरा तने खोए आउटर मोस्ट कवरिंग बची क्या सेल वॉल क्या हा गमा को ओजर आउटर मोस्ट कवरिंग बची क्या सेल वॉल फिलो का ची क्या कैप्सूल ठीक ना मेरा तने नंकु का जेनेटिक मटेरियल क्या जेनेटिक मटेरियल आर ऑफ generally are of two types ठीक ना मेरा they are either chromosome or plasmid या chromosome होत्या या plasmid होत्या तेने mesosome आकाने plasmid दुनला योगा स्थान ते दुगा मिनुक्ता इका sorry इका इंगे full screen चोस पासा इका nga आगा थोआ मेरा cursor बो है तेने mesosome दुगा मिनुक mesosome बो ची नुक जन्ना का तो they are इसुगो नंकु का projection है नुक mesosome गुने surface area बढ़ाई चो चेसे फिर try चो नुक तेने nga पोरिस गारे ATP synthesis or ATP synthesis chino ATP synthesize chokan ka wo energy currency of cells era da zara me thala ote gun ivi ka ma tene ote ve ka located yono tene nangku ayon sala tene nangku ote ve nangku chi ut kyanga ta cell membrane hala thik na mera cell membrane nangku cytoplasm thik na mera cytoplasm bo chi nok janna ka to cytoplasm is a semi liquid like substance that is present within that is bound by the plasma membrane that is bound by the plasma membrane ठीक है मेरा हागो मगो तेने ओते वे cytoplasm में तेने नंकु का the genetic materials which are chromosome and plasmid are floating ते उन्हें यहाँ पे चियोत organelles ngata ika basically चियोनो ribosomes योनो हागो मगो ता ई छंग में function बो nye जागो नजरन हे ठीक है मेरा ता ओते अगर ने डेमो चोस्ते यंगो माने इवा इन्ना वया ता मा सिंप्लिफाई चोन गाता है हागवा दक्सायं कॉम्प्लिकेटेड चिक दुगा मिनु खाला तेने काका क्रोमोसोम यत ठीक ना मेरा काका क्रोमोसोम यत हे प्लाज्मिड मन क्रोमोसोम ओते वांगा ता चीज रनो कोते जगह काका क्रोमोसोम काका लोकेटेड यत दैट एरिया इज कॉल्ड न्यूक्लियोइड ठीक ना मेरा चीज रनो न्यूक्लियोइड ओजे तेने सेल वॉल बची नंगे पगिरतो क्या म्यूरिन जकने ते आव नंगे पगिरतो क्या प्रोटीन नंग कार्बोहाइड्रेट्स नंगे प ओजर ठीक है मेरा ओते म्यूरिन जतन नक्ते बे फिया तेने ओते वे सेल वॉल फिलो का कैप्सूल दुखाला तेने ngata तेने ओते वे नंकु गची दु प्लाज्मिड ये चीज इसमें प्लाज्मिड इज वन ऑफ द जेनेटिक मटेरियल हाला present in the cytoplasm of the cell the other being chromosome tene ikane 70s ribosome to gamin iu changmangi chi chen daksa detail na zarin daksangi jagun cher macher iu chapik singe au chachik na chapik singa tang gwa changing nang ngerang nge jaguna ha gon kwari iu chi song basically ribosome ka be sub unit ni chon thala tene yang ngata particle yono gotyo depot substances metaphosphates se to ga iu nyang particle rikeno iu nge jagune label ma chona dik examiner and attachment pili du gaminu attachment pili is anodic sirf pili is anodic pili chi chon jana ka to it helps the bacteria to attach to the surfaces ठीक ना मेरा तेने ओते वे resgarek chi yondo resgarek nanku ka plasma membrane khato ka cell wall khato ka young membrane ji yondo khala ओते res mangchat pochi na yono gram negative bacteria na yono ठीक ना मेरा ओते membrane be ko protect chu chesna help jano ठीक ना मेरा hago mako tene onna choksla tene na next pochi du flagella chi na chu move help jano ko movement locomotion ठीक ना मेरा locomotion na help jano hey tene ngatang I vesting na ta chi utyak zen na ka to tanga tang ta daksangi chi lo taksarang ye gram negative ji spera tang sama tang gram negative spera tang chi nang gram positive ji yano ha goma go ote gram negative nang gram positive bacteria na agar nge farak postana ठीक ना मेरा ओते chi utyak zen na ka to the difference between gram negative and gram positive ठीक ना मेरा gram positive cells always appear purple ठीक ना मेरा हा गमा को gram negative pochi thono pink thono gram positive po purple ikane thong do thamino khala chip ya nok jana ka to chi jana gram positive cells ko ne ya cell wall bo mathi kyon ko peptide or glycan te layer bo ya mathi kyon gram negative ina chi yon jana ka to ote cell wall bo thin yon first secondly ngata daksarang iwe sangal namastas pinate gram negative cells have one more layer of membrane above the cell wall philoga cell wall philoga yang membrane jigyono khago mago ote be chi chonok jana ka to khwate purple stain bo nen jugaman nen nang khoi phangs tan ko ya tewe phe pink thon ko ojer pink po chi color duk ikanga ta stana ka to pink po khato ga membrane color bo to khala ठीक ना मेरा तेने ngata ta hago mago ta even ngata gram positive nang gram negative hala so ma song gram positive po chi utyak jana ka to yang go mane I repeat gram positive cells have thicker peptidoglycan I mean the cell wall layer cell wall bo peptidoglycan nang pugit to chaga was it gram positive cells have thicker cell walls and gram negative cells have thinner cell wall plus the gram negative cells have one additional membrane above the outside of the cell wall hala which doesn't let let the stain absorb ठीक ना मेरा तेवे पे ई खातोक न चिदुक पिंक थनो हा गो मागो tanga ta gram negative and gram positive in a difference ponga ta chichwen I mean we'll go into a bit of detail of what are gram positive and gram negative ठीक ना मेरा gram positive cells kun chin og jen ka to ote kun violet charukala purple ya violet jen na dik ba hala ठीक ना मेरा thick cell wall ठीक ना मेरा peptide and the thick peptidoglycan cell wall retains the primary stain bo chin yon ngata prime ngan la tangkan stain tanga tang daksangi yang detail na jere hey don't worry ote peptidoglycan chi zarano ngan la stain ngata ngis tano ote stain ba chi zarano ngata primary stain ba crystal violet chi zarano crystal violet te we koi violet color to gamino khala osit tene ote be jaste koi violet color bo songs pasang koi chi no ote koi pudama no sting na ote koi chi chonok longe puda manok iu itubur he puda manok I word pu idubur puda manok ozer tenna jo gram negative na chi chonok zen na ka to khoi otena sting na ngala primary stain espera ma sa gram positive na hala gram negative na secondary stain ta no ngala ngata primary stain bo chik ta sta iu yang detail na zen don't worry he yang yo tya next slides kunena gram negative na chi chanok ngala ngata crystal violet patano oteo shalanok hagomago tene ngata yang jiktanok hagomago yang stain jiktanok hagomago otewa ngata chizaranok safranin ya fushin hagomago otewa chizaranok safranin daksa chi itubor safranin itubor ठीक ना safranin e color bo pink one hagomago ta gram negative guna na ngata in ethanol nang po shalanok pa hagomago ठीक ना stains pink because of thinner cell wall gram negative cells have thinner peptidoglycan layer ngatanga figure na do gamino that allows the crystal violet to wash out on addition of ethanol nang yapong thus tang na ka to te a crystal violet po khoi shal te khons tano tenenge yang secondary stain bo chap na ching nang nga pink thono ha gomago ta gram positive na nga pink po absorb chonang violet po izuk strong color jigeno pe ki violet te pink po thong chu gamano ta oto that doesn't matter hagomago chi जना का तो ग्राम नेगेटिव सेल से chi चोनक जना का तो क्रिस्टल वायलेट पो ने जैसे गोलाक सिक चोक सिक नुक पाया हागोमागो गोलाक सिक ने पत इजुक सिक इथेनॉल बो साबुन जिक चोक सिक नुक हागोमागो गोलाक पो अगर माने थुकमेक चोक सिक ना का तो ओते काने दाग पांग छचेस का तो हमिने ओन्ना चोक सिक सामस्तो ओते क्रिस्टल वायलेट पो chi नुक दाग पनो थुकमो यतना का तो दाग पो बुदमानो श्रामो यतना का तो दाग पो बुचेस लामो छानो हागोमागो ote cell wall bo shamo yutna ka to tenenga ta crystal violet po butyanok ethanol nang po thu chiknang tenenga ta secondary stain butano sting na tangkan ne stain bo chi jananok secondary stain oteuchin safranin I mean I really I request you to remember this because this will most probably most probably come in your exam was it ठीक ना मेरा saffron bo secondary stain primary stain bo crystal violet tene chi nangya po wash chonok lo ethanol nangya po hala ta even gram bu chi ajana nok janna ka to khoi ive scientist ming bon kya Christian Graham hala khoi e technique po develop chos kya 1884 na thik na mera te phya khwa chi jara nok te phya khoi gya bam khoi mi kana taxta no khoi thik na mera tene te nangya po ta ngata Archaea spera tang sama tang hala Graham staining go bacteria na use chonok par Archaea na use choman ngata bacteria ये gram positive negative nang वैसे ngata mang pe ma chalak mang pe ha go man ok par te na chabik shik bacteria ha go ches phia yang chama you gram positive intik du gram negative intik du jete ta yang zumo bacteria ngi jutak a nang b hala chik chik jaste ngata infection song kya hala zumek song kya ote zumo chachi nang nang ne bacteria wo cha things te stana ka to ta zumo bacteria a nang b ngis mala a wo gram positive in kya b wo gram negative in kya so ma song b we ma disease mang po cause cha kya hala disease sok po cause cha kya so ma song tene bacteria cha things te agar nga pata chana you gram positive duk zen na ka to tene ta pakka bacteria b wo mantik raga tezu nga ta chabik shik ote chabik shik idea jik chan ya baki u mangpo ezu ge bacteria classification pe mangpe kol ngana manako you 1884 na jas pasang te jana bacteria gun ha khorang magwa no pami gune hala scientist guna te bi phe te jane chos na sen ya tene ote wasting nga ta agar nye ta microscope na ta nga ta you bacteria e gram positive na negative nga ta I mean, if you have, I don't know if you people have studied it in your class 11th and 12th, but if you can easily classify whether a bacteria is gram positive or negative under the light microscope. Light microscope is that I mean that is available in almost all the schools. Hala, ठीक ना मेरा colleges ना schools संगमा क्या मा टप्पो machine चिक मा नपा को हाला. Then ओते वे ना ची दुख जना का तो microscope ना पता जानो. So amasong ta you ngeja guna this is your assignment diring phia ya ki e lecture bo chap to sar testing na ki ing iske nang a nang b tu gaminok a nang b nang ngeja guna such ki gram positive ka onok gram negative ka onok jate hala ngechilo gram positive ka onok gram negative ka onok jate inna exercise po swa ta in the next lecture I'll tell you how to do this identify whether a bacteria is gram positive or negative e vesting nange shat tang naga to I mean the tene oteu dakse e assignment pe purpose po met kan chachera which is why I want you to do this assignment thik ra merak tene ngata we'll continue with the lecture tomorrow thank you so much tene nge jaguna if you have any doubt just all of you know that I'm just a call away thik ra merak nga just nam call chunang dik hagama माने हेजिटेट माछा yang ngan la ngeja gune college asil kan jege thuguri gyot na ka to tis ki sir da dur ka juk sigot jate khawa ke jaz mar do chamet agar ngeja gune man kan jik chona mane he ठीक ना मेरा tene chi doubt yot na tis bacteria ko la chi daksa diring e exercise po agar iwe diring mati sting na iwe ko la ti na dik he daksa ngira exercise po you microscope na ngata stas no ki bacteria sample ni stas kya चिक चिक पे कलर वो ए चोक्स था क्या चिक चिक पे कलर वो बी चोक्स था क्या ए उची दुक पिंक दुक बी वो वायलेट दुक तांगे होते हैं आशातन ठीक है मेरा even ang nangeja guna you just have to tell me that which one is gram positive and which one is gram negative bacteria hago mago thank you so much tene daksa tene yang next lecture bo thori I wang ring mo song ya par nge try chun ki man nge na mera thik mera tene let's see you in the next lecture thank you so much